the day before and also a quick preview of what we're planning on doing that day. So I've started the recording. Today's lesson is all about enzymes, but we're also going to have a conversation about the relationship between nucleic acids and proteins at the beginning of the lesson. Today's date is January 15th, 2021. And we've got two objectives and an essential question today. We use these objectives and the essential question as a guiding light. They give us a goal, they give us a baseline. They give us something that we hope to achieve by the end of the lesson, and they also give us a way to look back and answer a simple yes or no question. Did you learn what you set out to learn? You can also use these objectives and essential questions at the beginning of your notes, just to remind you when you come back and you're studying what exactly we went over in that lesson. Okay, so there's good information to write down on this page. Definitely the title, definitely the date. And if you choose to, the objective and the essential question are probably good ideas as well. But the first objective today is to explain how enzymes act as catalysts for biological reactions. The second objective is to explain how the environment or an environment can impact an enzyme's ability to function. So hopefully you all have done the assignment from yesterday, the asynchronous assignment from day 3. One of them was about protein research, and I asked you to look up on various websites 5 different types of proteins. One of those proteins was called an enzyme, and we're going to talk a lot more about enzymes today. But hopefully, based on the research that you did yesterday and the summary that you typed up in that asynchronous assignment, you have a good starting point for this conversation. And then lastly, the essential question, what does it mean for enzymes to have an optimal environment? Okay, so I want to return to this chart. We introduced this table yesterday because I believe that if you can memorize this table and have a good understanding of the 4 parts that make up this table, the 4 columns, then you are in a great place to succeed in Unit 1, and you have established a really strong foundation to move forward in our course. So we have those 4 biological compounds, which are in that far left column: carbs, lipids, nucleic acids, and proteins. On Wednesday, we introduced carbs and lipids, and yesterday, on Thursday, we introduced nucleic acids and proteins. And in each case, I told you all that there were 4 things that you needed to know about each biological compound. You needed to know the monomers that make up each compound. You needed to know the elements that make up each compound. You needed to know the compound's function, and you also needed to be able to provide examples of that compound. So we did the first 2 rows yesterday. And then yesterday, we also introduced the last 2 rows. So at this point, we should be able to fill out this entire table. So that's what I want to do. Can someone tell me what is under box 1? Which monomer makes up carbohydrates? You can tell me in the chat, or you can tell me.
Monosaccharide.
Good. And Rudy, thank you. I appreciate it. You said glucose in the chat. We can say either one. A glucose is a monosaccharide. What about under box 2? What elements make up a carb? And let's get as many people participating as possible. If you don't know, then you should be writing along with us. What elements make up a carbohydrate?
Carbon, hydrogen, and oxygen.
And what's special about those elements when it comes to a carbohydrate?
Their ratio.
The ratio, good. There's a 1 to 2 to 1 ratio. So we did this exact thing yesterday. So hopefully everybody has access to the information. So I want you all to start sharing out more. We hear a lot from Kathy. We hear a good amount from Brianna. We hear a good amount from Rudy. Let's get some new voices participating today. What's the function of a carbohydrate? What does a carbohydrate do for a living organism? Excellent. Thank you, Rudy. So they provide quick energy as well as structure to the cell. What are some examples of carbohydrates? Yeah, and thank you, Breon, too. I appreciate it. What are some examples of carbs? Sucrose is a good one, Jason. Thank you. And he brings up a good point as well. When we've got a word that ends in OSE, that should be telling us we're talking about a sugar. We're talking about a carbohydrate. So can we think of any other words that end in OSE? Fructose, good. Then we also introduced cellulose, galactose, excellent. We're getting a lot now. And then glycogen and starch are also examples. They don't end in OSE, but they are still carbohydrates. Okay, next row. So let's move through this one quickly. What's the monomer that makes up a lipid? It's kind of awkward to say. What is the monomer that makes up a lipid?
Glycerol and fatty acid.
Good, thank you. Glycerol and 3 fatty acids. And thank you in the chat, Jason. I appreciate it. And what about the elements? Well, I'm just going to give you guys this one because they're the same elements that appear in all 4 biological compounds: carbon, hydrogen, and oxygen. What's the function of a lipid? And thank you, Michaela, for your answer in the group chat. What's the function of a lipid? They store energy. Yeah, they provide long-term energy. And they also provide some insulation. I think that was something that someone said yesterday. It's not here, but it is true. Lipids provide insulation and they make up the cell membrane. So again, hopefully if you were not here yesterday, or if you, for whatever reason, didn't write down this table, that you're taking time to write it now. And lastly, what are examples of lipids? Butter is an actual food example. That's a good one. Oil is another one. Thank you, Brianna. Waxes, excellent. We also introduced steroids. There are some steroids that are lipid-based, and then phospholipids is a good word. I think someone sent that in the chat yesterday. I was impressed. Okay, the next row, nucleic acids. So this is what we introduced yesterday. And if you created this table in your notes yesterday, then you need to go ahead and add on these next 2 rows. The nucleic acids, what's behind box number 9? What's the monomer of a nucleic acid? What is the monomer of a nucleic acid? We talked about it yesterday. Nucleotides. Thank you, Jason. And nucleotides have those 3 parts: a 5-carbon sugar, a phosphate group, and a nitrogen base. What about the elements that make up nucleotides? Because we've got those same 3, carbon, hydrogen, and oxygen, but we've got a couple others as well. What are the elements? We've got those same 3, carbon, hydrogen, and oxygen, but then there were 2 others that we introduced.
Nitrogen.
Nitrogen is 1. Thank you. And there's one more.
It starts with a P, but I don't know how to pronounce it.
Thank you, Kathy. Not
Oh,
prosperous. Phosphorus. Phosphorus. And the abbreviation for the element phosphorus is just P. I didn't type that in, but it's P. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay. What is the function of nucleic acids? What do they do for living organisms?
they gave us DNA.
Yeah, DNA is one of the examples of nucleic acids. So really what we say is that
Could take genetic information.
they contain the genetic information. Let me type that in specifically. Okay. And then the examples, Kathy already gave us one, but the other is RNA. So DNA and RNA. So I'm going to leave this up right now for a couple more seconds before we go over proteins. Make sure that you do have this table somewhere where you can refer back to it. Okay, proteins.
Amino acid.
Good, thank you. So the monomer of a protein is an amino acid. There are 20 amino acids. You do not have to memorize them, but it's just good to know that there are 20 because that leads to so many possible combinations. What elements make up proteins? What are the elements of a protein? Thank you, Rudy. Carbon, hydrogen, oxygen, and nitrogen. Two more boxes. Let's go ahead and knock it out. What's the function? Well, they do a lot. They're the do-it-all compounds in the cell, so I won't make you guys kind of guess around that. And can we think of any examples of proteins? Again, I asked you guys to do that protein synthesis, protein research assignment yesterday. So do we know any
Enzymes.
enzymes or other primary example? Any others that we could think of? Antibodies, and then there are also hormones, insulin, and hemoglobin is another one that we talk about. So I'm going to leave this up for 90 seconds. 90 seconds. Please make sure you have it written down. You have a screenshot. You've taken a picture of the screen. Whatever you need to do to have access to this, it's going to go a long way. Trust me. Just about 30 seconds left. Let's move on. All right, so take a look at this mind map or this flow chart. This is another way that we can create a visual representation of these 4 organic compounds. Now, based on your notes, I want you to ask yourself a question. Could I fill this out? And hopefully the answer is yes, because we got all of the information that you need to fill this out written on in that last chart that was just on the screen. But if the answer is no, then obviously you have more work to do and you need to pay closer attention and watch some of the previous lessons and take notes. I don't want you guys to get behind in the class, and this is really, really important foundational information. So it's so important and foundational that we're going to go ahead and do this mind map. As you can see, we've got at the top our 4 organic compounds, and in each chain, there is some information that's already been filled out to give you a hint as to which organic compound is where, where we place each organic compound. So for example, in this first chain, it says blank, which are made of amino acids. So what is made of amino acids?
Protein.
Good, thank you, Kathy. Proteins are the organic compounds that are made of amino acids. So organic compounds in living organisms include proteins, which are made of amino acids. Examples include antibodies, and then we also included we talked about some other examples. You don't need to write this down, Brianna, but this is maybe something else that you take a screenshot of, or you could write it down. It's up to you. It's just another way of representing the information. Good question. And what are some other examples that we just talked about of proteins? There were enzymes. Thank you, Michaela. What else? Hormones, good. And then I also included hemoglobin and insulin. Okay, let's look at this second stream or second chain. Here, we're not really given much information until that last circle. It says also known as. So we're thinking of blank, which are made of something, which could also be known as cellulose. So what do we think this stream is talking about? Which of our 4 organic compounds? Good, excellent, Jason. So this is about carbs. So carbohydrates, which are made of, what's the monomer of a carbohydrate?
Wouldn't it be glucose?
It could be glucose. I have another word here. What's the other there it is. Jason in the chat says monosaccharides, which could also be known as Abraham just said, glucose. And Michaela said it in the chat as well. Okay. Then we're told that lipids go in this third stream. Organic compounds in living organisms include lipids, which are made of what?
Glycerol.
Glycerol and 3 fatty acids. And whose jobs include what?
To provide. To provide Yeah,
Long-term energy.
Long-term energy, good, thank you. And I think, Kathy, that's what you were on the verge of saying, and Jason said it in the chat as well. And there's one other thing that they provide for us, or a couple of other things. Give me one of them.
is it the cell wall? Oh yeah.
Cell membrane, and we're going to talk about the difference between those two. But Jason also says insulation, and that's important. So I like to think about lipids as being those fats, and I like to think about it in terms of a polar bear or a walrus. They have a lot of fat to insulate their bodies, to keep the heat in. Okay, so lipids are needed in cold environments to keep heat in. That's called insulation. And then that only leaves one other organic compound. So what goes in that last stream over here? It says two main types are RNA and blank. So what should go over here in this last stream? Nucleic acids, good. Which are made of what? What's the monomer?
Is it like amino acids?
So we've got amino acids for proteins already.
Nucleotides.
Nucleotides, good, thank you. Someone said it. What's the other type? So if RNA is one of the types, then what's the other one?
DNA.
DNA, thank you, Jason, Rudy, and Kathy. And these two things will eventually lead to proteins. So that's kind of the relationship that we're going to talk about coming up. Nucleic acids eventually lead to proteins. So they're very, very closely related. Good job on that. I can leave this up for about 30 seconds if folks want to try to write it or take a picture of it. And yeah, Jason, so you're right. It does lead to genes, and genes eventually lead to proteins. So every gene codes for a specific protein. So you are correct. I just kind of skipped that step, but thank you for mentioning that. Okay. And this is what you're talking about. So genes are small segments of DNA found on a chromosome that code for proteins. So human DNA, for example, is about 3 billion base pairs long. So what that means is that those A's and the T's and the C's and the G's, if we were to line them up, so it's A, T, C, T, T, A, C, G, A, G, G, A, T, all the way, 3 billion of those. A gene might only be a section of a couple hundred. And so in the human DNA, we've got 3 billion of those A's, T's, C's, and G's, and we've got about 30,000 genes along the way. And a gene is just a small section of DNA. And each of those genes is going to code for between 1 and 5 proteins. So this is the essential relationship between DNA and proteins. DNA has genes, or it makes up genes, and those genes then code for proteins. Okay. So you don't need to write this down right now. We're going to go into this in much greater detail in Unit 5, which will come up probably about a month and a half from now, right before the midterm. So for now, what I just want you guys to see is the big idea, the relationship between these terms. Nucleotides are the monomers that combine to form nucleic acid, specifically DNA. And then DNA gets converted into RNA through a process called transcription. Again, you don't need to write this down. We will talk about it in much greater detail. Eventually, that RNA is going to signal for certain amino acids, and those amino acids will combine to form proteins. So this is a cool process. It's called protein synthesis. You start off with DNA, and eventually you can form proteins. And proteins are the basis of our traits. Proteins make us who we are. They give us our physical characteristics. Here's that relationship one more time. And this entire time, it's really the sequence of things that is important, the way that they are ordered. So the sequence of the nucleotides matters. ATC is different than TAC. GGC is different than GCG. When you switch the order of those nucleotides, you're changing the DNA sequence, and eventually you could have an impact on the protein that's formed. So the order of the nitrogen bases determines which amino acids will be used, and also their order. And then the order of amino acids determines the shape and function of a protein. Now, enzymes are proteins. So this is stuff that I would like for you to write down. This slide right here. This is not important right now. We're going to go into much greater detail about this later in the course. But this is what I do want you to write down. Enzymes are a class of proteins that carry out various functions in living organisms. The primary function of an enzyme is to increase the rate of a chemical reaction. And when we say increase the rate, what we're talking about is speeding things up. Enzymes speed things up. And this is why we call them catalysts. A catalyst is something that speeds up a process. Every enzyme has a unique shape. And that unique shape is determined by the sequence of its amino acids and also how it folds. So we can see that folding process happening here. It kind of starts off as this chain, but then it specifically folds into a unique shape. So it starts off as a chain of amino acids, and the chemical properties of those amino acids will determine how the enzyme folds. Okay, I'm leaving this up for about 30 seconds. So it sounds kind of corny, but the way that I used to remember this when I was in high school, the Z in enzymes always made me think of like the word zoom, something that moves fast. So an enzyme zooms the chemical reactions. It speeds them up. It helps them to go fast. And for those of you who are interested in medicine, or you might think you might become a doctor or a nurse or a physician's assistant, these enzymes are so crucial to the body because they allow things to happen quickly. Processes that would otherwise take forever, not literally forever, but that would take hours, can now be done in seconds or in minutes. For example, we need to be able to break down food when we are digesting it so that we have instant access to the energy that is stored in that food. Enzymes allow us to do that. Without those enzymes, we would have to wait a much longer period of time to actually have access to that energy. So enzymes are pretty cool. They have this process called, or they have this quality called specificity. Enzymes are specific. And that means that they can only act on substrates that have a specific shape. So as you can see in this cute little image here, enzymes will only fit with a substrate that is shaped similarly to their opening. Okay, now there are some important terms here that we're introducing. So the substrate is the reactant in a chemical reaction. It's the thing that is actually being acted upon. The enzyme is going to do something to the substrate. It might break the substrate down, or it might take the substrate and combine it with something else to synthesize something, to make it, to build something. But that substrate is the reactant. It's being put into the reaction, and something's going to happen to it. Every enzyme has what's called an active site. Typically, the active site kind of looks like the mouth of a Pac-Man, and this is where the reaction takes place. So this is where the substrate actually gets acted upon. It's where it gets broken down or it gets built up, whatever that enzyme's job is. But this specificity is so important because it makes things like a lock and key. The keys that I have to open my apartment will not allow me to get into your house. They don't allow me to get into West Mech. They don't allow me to even get into my own car. They work only for one door, and that door gets me into my apartment. So enzymes are the same way. They can only work with specific substrates. And this specificity is guaranteed by the shape of something. So it's not about, oh, they were just in the same place at the same time. It's not about they were produced by the same cell. It's all about their shape. So the substrate has to be the same shape of the active site. So let's take a look at this. Which of the following enzymes is able to act on substrate G below? So here's substrate G. Pay close attention to how it's shaped. So which of the following enzymes is able to act on that substrate? Which one of these enzymes that I'm about to show is actually going to fit with the shape of substrate G? Which enzyme will fit with substrate G? Cindy, did you have something to say? I thought you came off of mute. Okay, maybe not, but we've got several people in the chat, Jason, Rudy, Michaela, and Abraham, all saying enzyme 1, and you are correct. So we can see that the substrate clearly fits into the active site of enzyme 1. So enzyme 2 has an active site that is shaped like an arrow. So it's not going to fit with substrate G. They don't go together. Enzyme 3 has an active site that is shaped like a wave of some sort. It's not going to fit with substrate G. Only enzyme 1 is going to be able to act on this substrate. Okay, so this is something that I think would be a good idea to draw. Again, visualization is important. I can almost guarantee you will have a question like this on almost every test that we will take this year. You will be asked to match the parts of a diagram that looks a lot like this. So what are the substrates? What's the enzyme? What's the active site? What are the products? So remember, your substrates are what actually go into the reaction. They start off in the reaction. Your product or your products are what come out of that reaction. They are what is produced, or what are produced.
Do we draw this?
Yes, I would like for you to draw this. It doesn't need to be perfect, but 90 seconds. Okay. Did my timer go off? Yeah. Not yet. There it is. One other thing that I'd like to point out while you all finish up your drawings, if you look at this enzyme, it does not change throughout this process. So the enzyme starts off exactly how it ends up. Now the reactants, the substrates, they do change. They start off as one thing and then they get broken apart. The enzyme breaks them apart. That's the chemical reaction that happens. But the enzyme itself does not change. This is a very, very important quality. Because what this means is that enzymes are reusable. We can use an enzyme and it's still going to be usable at the end of the reaction. I can do the exact same reaction with different substrates because it has not changed shapes. So enzymes are specific, and enzymes are catalysts, and enzymes are reusable. Okay. So technically, there is not an asynchronous assignment. There shouldn't be an asynchronous assignment today. Yeah, this is not going to. I'm unpublishing that. So you can go ahead and choose to get ahead and do the Amoeba Sisters enzyme video and quiz for Tuesday. That's really supposed to be done on Tuesday. You can try to do it today, or you can go back and make sure you get some of your missing work done that you've gotten from earlier in the week. We do have an exit ticket. but everybody has something to do. You can choose to get ahead or you can choose to catch up, depending on what your unique circumstances are. All right, so I'll be on. What's that?
Where's the video and quiz at?
It's called U1D5, Amoeba Sisters, Enzyme, Video, and Quiz. It's on Canvas. Yep, we've got 21 minutes left, so I'll be here on mute. Let me know if you have any other questions. Thank you for that question, Kathy. Okay, folks, it is now 11:35, so you've got 30 minutes to enjoy a lunch break before a third block. I know you need to push through these last two blocks to get to the long weekend, and I will talk to you all Tuesday. If you need anything between now and then, do not hesitate to reach out. I'm proud of everybody for working hard this week and for keeping their attendance up. So everybody have a good weekend, and I'll talk to you all Tuesday.